announcements for the week. So Thursday you have an in-class exam, right? Tomorrow we're going to be doing a discussion based on phylogenetics. So that's what we're going to lecture today. We're going to do practice tomorrow, and then you're going to be tested on it on Thursday. So it's going to come really quickly. So I would highly suggest to look at the discussion questions and try them. I think it's a fun activity, building phylogenetic trees. Dr. Gruey proposed it. It's something she used to teach a lot at the University of Iowa when she was there. It can take a decent amount of time to try it beforehand, because the other thing we want to do tomorrow in discussion is to go over the study guide if you have any questions, right? It's going to really get you experience with phylogenetics as new material, but also review some of the stuff that you'll be expected to think about on the exam. Related to that, I graded solution strategies pretty harshly this last week. I know for several weeks I was grading them kind of leniently. They were kind of straightforward, like do this, do this, do this. Last week it was super obvious that the majority of you did not think on the problems because you showed up in the discussion like, I don't know what I'm doing. And then I looked at the solution strategies and it's like, I don't know what I'm doing, right? That's not going to bode well for this exam. So please, please, please look and try the discussion questions for tomorrow ahead of time. Don't just show up and expect to get it done. It's not going to work. Okay, so today we're going to cover phylogenetics, right? There is genetics in the name. So what does the philo part come from? The philo part is thinking about relationships between different things. It can be lineages, populations, cells in your body. It's really a technique to think about relatedness or how similar things are to one another. So I've drawn just really quickly like a phylogeny, right? Call it a cladogram for context, and name those context. Well, how we're going to build them in a genetic sense is thinking about molecular markers. You can build these based on phenotypic traits, and we'll do that to kind of show the principles of parsimony. That's a really important thing on phylogenetics, but at the end of the day, we really care about these molecular markers. So why are phylogenetics kind of important in the context, the broader context of genetics and evolutionary genetics? You see it a lot in conservation. So this is a map of different chimpanzee populations. Are they the same population? Are they different populations? Are they the same species? How would you determine relatedness amongst these chimpanzee populations? You can take pedigrees. The pedigree and phylogeny are actually really similar, except the pedigree we know the relationships, the phylogeny we're trying to infer the relationships. But these, there's a lot of examples like this, where you're trying to understand the relationships between extant, like existing populations. You see this type of analogy, like this type of analysis, happen a lot in cancer genetics, right? This is, I would argue, it's very similar to a phylogeny, right? You're tracing cell lineages through development and thinking about the cells that are more closely related or come from a common source that would end up giving you cancer. So this would be kind of like a clonal analysis in the terminology of cancer genetics. It also tells us a lot about our own evolutionary history. So I talked a little bit last time about genetic tests. It seemed like some of you were thinking about them, or some of you had some experience with genetic tests. And I mentioned that, we as humans, some of us, depending on your kind of background, might have the kind of lingering traits related to Neanderthal gene flow. So how is that determined? How is it determined that you have nucleotides in your genome that are coming from Neanderthal? It's from kind of this type of analysis where you're looking at DNA sequence and looking for relationships, right? So if you have a SNP of genes, you can draw a tree of relatedness, right? But maybe there's this one variant, so each tip represents a sample, right? And then the relationships are represented by these branching effects. Maybe you have something way up here that you don't know what it is. It looks really different from any of the samples that are in most modern human populations. How would you determine that's Neanderthal, right? You have to add something. You have to kind of add genome sequencing, and then you tell that these two samples are more close related than kind of the rest of the human samples, right? Human is a weird word to use. They're all hominid, but hopefully you get what I'm saying. So relationships like this can be inferred from genomic sequences. So I think I threw some of these stats at you last time, but for someone with Eurasian, or like so European Asian descent, right, about 2 to 4% the genome looks like this pattern, where your SNPs would cluster more closely to Neanderthal samples compared to other alleles in modern human populations. And this was all accomplished by the Neanderthal genome. This was in the journal Science in 2010. Right? Before that, human geneticists didn't know where these SNPs were coming from. They had a very distinct pattern. You could guess that they were coming from some ancient introgression, but the real proof came when we had something to compare it to, right? That's what phylogenetics is all about. It's not about unique differences, it's about shared differences. You can't do anything in phylogenetics with kind of unique things. You need shared commonalities, and we'll get that. So what do these traits look like? Some of them are linked to hair density, male density, skin color, but there's some really interesting ones, including reduced fertility. And that seems to be X-linked. A lot of the DNA that might be still in populations of Neanderthals on X. Why? X undergoes less recombination, right? It only undergoes recombination in XX individuals. Has a different effective population size. We didn't talk about that in population genetics, but effective population size is really, really important. So it's really interesting that a lot of these traits are X-linked. It's also really interesting that this other aspect of X-linked, it's very unlikely that the type of introgression that occurred in ancient lineages was Neanderthal XY, human XX. There's no Y. We haven't seen in human populations anything that looks like a Neanderthal Y. So it looks like introgression is also asymmetric. How else does phylogenetics affect our health? Does anyone know what this is a picture of? It was really, this picture was all over the news like last spring. Yeah, it's a genealogy of COVID strains, right? We can tell where different strains of COVID are arising based on their sequence similarity to strains we know about. There still is a big argument, where did COVID originate? It doesn't look like, you can test the hypothesis whether it was from kind of a market or a lab by making different biology. It looks like it was most closely related to samples that are segregated and not in the lab, right, in market parts of China. When new variants come around, you can put them on a phylogen. Where did Omicron come from, right? South Africa, because it's more closely related to other strains in South Africa. You can see recombination events. So this new variant that's floating around that gave all of you guys gastritis last week, you can see that it kind of is in between strains. So you can infer a ton of stuff through phylogenetics. But at the base of it, what we want to understand is evolution. So we did population genetics to understand contemporary evolution, like evolution as it's happening. This is more at a larger scale, right? Evolution that's happening between populations. So we were defining evolution as the change in allele frequency over time, and eventually it gives you kind of this splitting event. So your book defines kind of cladogenesis and anagenesis. You don't have to remember those terms, but I think it's really important to point out that if you're seeing a phylogeny, it's not that evolution is just a splitting event. Evolution is happening this whole time, right? Often there's a misconception that if you see on a phylogeny like a long branch without splits, that it's not evolving. That's not true. You'll see people refer to things as basal. That's a horrible term. There's no such thing as basal in a phylogeny. Everything's evolving. The length of the branch in a tree like this tells you the number of mutations, but they're all evolving. The length of a branch here doesn't tell you about evolution. It's just trying to give you an aspect of time that order these splitting events. So if phylogenetics is all dependent on evolution at a population scale, then being partitioned, what does it rely on? It relies on there being genetic variation, right? Same with population genetics. You can't have evolution without genetic variation. So what does this variation look like, right? do we think this variation is going to look? It's out there. What is it? What are some characteristics that we can describe genetic variation? Okay, we'll go to the clicker question and we'll come back. So this was a point that your book was making about genetic variation. So the question is, which statement is true about the neutral mutation hypothesis? All proteins are functionless. Natural selection plays no role in evolution. Most molecular variants are functionally equivalent. All of the above. So as you're answering this, let's go back. What does genetic variation look like within a population? There's a lot of it, but how would we characterize it? Sure, we can think about phenotypic differences. What about at the molecular level? Yeah, they are mutations, and what's the effect of the mutation? They're mutations that we can observe, but they don't necessarily have a functional consequence. So that's the answer. That's the best answer to the clicker question, right? Most molecular variants are functionally equivalent. We've seen codons. It's really only if you change certain parts of a codon that you change the amino acid. Introns, who knows? As long as you're not changing splice variants, probably nothing. And then we've also seen intragenic regions, right? Most of the human genome is not coding for a gene. Maybe it has function, maybe it doesn't. A lot of the mutations, you're never going to see a functional consequence. You're never going to see that phenotypic consequence because they're functionally equivalent, or because they're in DNA that currently you don't think of as functional. So mutations, most are going to be silent. The other really important part here for you to know is recombination. We've been thinking about recombination often in terms of doing crosses and recombining things to think of physical maps, but that's always happening too. It's making new variants feed together, right? Recombination can help you with pedigree. It can also help you here in phylogeny, thinking about how new variants arise, right? Most mutations arise that are rare, they're recombined on different backgrounds, and they spread through recombination. So the best answer here is C. Natural selection is one force that plays a role in evolution. That's how we're going to. This, and then A is just too much hyperbole, right? Not all proteins are functional. So that also rules out B. So that neutral mutation hypothesis kind of recapitulates that, what I just said, right? So most mutation has little effect on survival. I'll say on fitness. So it's segregated. It's not going to be selected out of a population. It's not going to contribute to differences in mating. It's drift, maybe depending on the population size, but if the mutation's occurring, right, we know something about equilibrium due to mutation. It's just due to that mutation rate. As long as there's no selection, this allele can keep accumulating, and then you can think about the spatial structure, the lineage-specific structure of these mutations, right? So this is what they're calling the neutral mutation hypothesis. You can test this hypothesis. How would you test this? Chi-square. Here's my frequency that I expect at equilibrium. Here's the frequency I observe in a population. Is it different? Observed and expected, right? So how are we going to look at variation, molecular variation? We're not going to see it in this lecture, but we'll see it next week when we start getting to techniques. You can sequence DNA. You can use things called restriction enzymes. So those are enzymes that cut specific DNA. So if there's SNPs, if there's differences in alleles, you might have different cutting patterns. You can use microsatellites. Those are sequences that expand due to kind of runs in repetitive sequence, so they have a distinct size. These are super, there's a lot of allelic variation in microsatellites. They're a really good tool. Now there's some older stuff too, like people used to use protein size or shape. The ADF-H alleles you saw in Drosophila, the F and S stand for fast and slow. That's running them on an allozyme gel, how fast they run down a gel. So that's related to size. So there's a lot of examples in your book of how you can think about connecting DNA sequence variation to population differentiation. We'll see a little bit of that next time. So now we're going to start thinking more about these phylogenies. How do we build them? How do we read them? Right? There's a lot of information stored in them. How do we kind of make that algorithm for thinking about how we're going to deal with this? So this one is actually called a gene tree. So that's providing information about the evolutionary relationships among genes. So these are just examples right now, just to get you thinking about kind of where we're going and what these are looking like. Does anyone notice something about this tree? Yeah, some lineages are listed multiple times, like crocodile, PRL1, PRL2. So those are called duplications. We've seen duplications. Duplications are going to be important in phylogenetics, right? So when we have one of those. So when you're thinking about a phylogeny at the gene level, so we can even zoom in within a gene, right? We saw a phylogeny of gene genealogy of COVID sequences. This is kind of the next level up. So all of those COVID sequences we saw kind of in the very first slide were all samples from human. Now we're just taking a single gene and looking at samples across different animals, right? There's different duplications. Those can be really informative when duplication occurs, how we're going to measure molecular changes. Some of the relationships between animals you wouldn't necessarily expect based on what we think relationships between animals are. So the other point I want to make about gene trees is that, we often think of like there's one phylogeny, right? We'll see an example in the next slide. There's only one way that we're related, but every phylogeny is actually more of a hypothesis. I hypothesize that certain sequences are related in this manner. So what's really interesting is that even though we'll see a trio later, like human and chimp were most closely related, and the next one is gorilla, even though that is the relationship that we think is pretty robust, gene trees do not always have to look the same across the genome. In some parts of our genome, humans are more closely related to gorilla. In other parts, they're more closely related to chimp. So even though there might be one true species tree, you haven't observed that, right? That happened so far in the past that we didn't observe it happen. So each phylogeny is a hypothesis, and that's why you can get gene trees that are different within even the same lineages, because individual genes have their own evolutionary history. But this is kind of what you're more used to seeing, right? You're more likely used to, and depending on what kind of intro bio you've taken, seeing something like this, right? Where we think that there's one kind of definitive species tree. So this would be called either a species tree Some people, there's too many E's, some people would call this phylogenetic tree. I think this phylogenetics is kind of the more overarching term, and species tree, gene tree is kind of the more specialized term. But I think in your book, they really, when they go kind of the next level out in terms of diversification, they're using this term phylogenetic tree a lot. So what is it? It's a graphical representation, but it has important information. We're going to start defining some of these terms, right? It has branches. It has nodes. What are these nodes representing? I can sort of hear you. Yeah, so that's the really interesting thing about these nodes. It's a bifurcation, right? It represents a splitting event, like a speciation event, but it also represents something else. It also represents a most recent common ancestor. So if we're thinking forward in time, we're seeing a split. But if we're going backwards in time, it's identifying commonality. That's going to be super important for building phylogeny. A lot of our terms, monophyletic, paraphyletic, whatever phyletic, are based on whether we can identify these common ancestors. So branches, excuse me, sometimes branches can show the rate of evolution, like if they're scaled by a clock or something. Here it's not. But here, what's really important is that each lineage has equal branch length. And then we have these kind of things here, tips. So each lineage is represented by a tip. So nodes, branches, tips. Any questions on this? There's a lot of slides, so I'm going to go kind of fast, so please stop me if there's questions. Yeah.
What is it like when it like breaks and breaking the line at the top of the course? What does that mean?
Yeah, that's a really good question. This is what people use in math. Like anytime you're seeing, I'm trying to think of a good example. It represents that the line is much, much longer, but if we were trying to show the true length of the line, it'd be hard to visualize, so we just put a line break. So you might have seen that on like a graph where it's like in millions. Put a line break because you want to kind of condense, and that's what it's showing here. So that's a really good question. It doesn't have anything functional, but if you drew this to scale, like how far away horses are from zebras, you could fit it in a picture. So I don't know what the term for that is. I call them line breaks, but yeah, that's good to notice. So you're reading, there's a ton of reading about what is a species. I think that question is really interesting, well, from kind of a biological and a philosophical thing, but we don't have time to go over that. So I think also in your homework questions, achieved, hopefully that reflected that, right? I'm not going to test you on allopatry, parapatry, premating isolation, post-zygotic isolation. I didn't test you that in the reading. We're not going to cover that. It is important, though, to think about what is a species. If we're trying to make relationships between species or identify, this gene comes from this species, we do at some level have to have a working definition of the species. And the most common one that's used is called the biological species concept. So that's a species, is a group of organisms that can interbreed and they're isolated, reproductively isolated, so not exchanging genes with other species. So that reproductive isolation part is the really hard part. sometimes that's really hard to prove. Sometimes that breaks down, but we still kind of have these ideas of species, right? Like a horse and a donkey are species. They can mate, they can make a mule, that mule is sterile. So maybe they are reproductively isolated. They're not isolated in the terms that they can't mate, but there's not going to be gene flow because of the sterility of that one individual. What about the fossil record? People call different fossil species. We can't test reproductive isolation in a fossil. So this is the best kind of concept that we can have going forward, but it still has its limitations. All concepts have its limitations. Right? Some species don't reproduce sexually. What about bacteria? How are you going to apply this concept to bacteria? So it is problematic, but it is the one that I want to think of when we're kind of defining a species moving forward. So how are new species formed, right? We're going to infer the relationship from molecular data, but there has to be something kind of happening behind the scenes that's going to generate these really specific patterns. So I'm just going to quickly go through allopatric differentiation, allopatric speciation. There's so many different examples in the book. They're really interesting, but this one is the most easy, I think, to wrap your head around in terms of how it would lead to kind of a bifurcation. You have an ancestral population in green, then a mountain just explodes in the middle of Nebraska, and it separates the populations. They're independent now. They're kind of going on their own evolutionary trajectories, not exchanging genes. They accumulate differences, so they become blue and red over time. And if you hypothetically brought them back together, they couldn't mate with each other. Or they could, but they would produce non-viable or sterile offspring. So that's kind of an example of how a vicariance event, this geographic vicariance, would lead to a reduction in gene flow, independent evolution, and the evolution of reproductive isolation. So here's the next top hat question. What do you think will happen if migration is taking place? Yeah, there's this mountain, but 10% of individuals fly over the mountain every generation. So I'm meaning something like this. They're continually exchanging migrants. Migration coefficient is 0.1. That's what I said verbally, 10% being exchanged. This is reciprocal. Yeah, so there's a lot of really good, interesting questions in here. I think the short answer is there probably wouldn't be a speciation event. They continually exchanging genes, they both look green at the bottom. That's not 100% true. There could be other forces leading to speciation. So I was just trying to get you to think. I don't think there's any best answer, except that you're thinking about gene flow, you're thinking about hopefully making connections to last lecture where we said migration homogenizes things, right? Even say that the gene flow didn't happen until this stage. Purple, red, and blue, you homogenize them. They wouldn't be species anymore. It would kind of just be populations that live on two different sides of the mountain. Okay, I'm not going to go through these, but these are like all the different ways that you can stop gene flow. It's really interesting that not all of them are like active processes, right? Temporal isolation. This happens a lot due to phenology. So if you're a flower that blooms in May, and your close relative blooms in June, you might not have changed genes just because of that. It's not anything to do with like not recognizing mates or making progeny that are sterile, just bloom at a different time. So back to this figure, we have all those things reducing, kind of acting as barriers, but what's causing the differentiation within a population? So geographic barriers or mating barriers are keeping them apart, but what's causing them to change color? A lot of good answers. It's all the things, right? This is everything you saw last week. Inbreeding, you don't have to memorize any of these equations, right? You have to know the concepts. So what's causing these populations to change color? Evolution. All of the forces that we talked about. Inbreeding, right? You're from a big population, now you're kind of this isolated population on two sides of a mountain. Mutation, maybe migration, we don't know. Drift is definitely happening. It's a big population, we made it smaller. Drift is stronger and smaller populations due to that big N, total number of chromosomes being on the denominator. Natural selection, fitness, mutation, and selection can reach certain equilibrium. So those are all the different things you saw last week. Those are all the processes that are contributing to genetic variation or differentiation in technology. Okay. But back to these trees. What do we want to answer? What do we want to know about these trees to be able to interpret them? We want to start building that kind of algorithm. This is the first thing I'm going to look for. This is the second thing, third thing I'm going to look for. So the big question we want to ask are, how closely related are lineages? I kind of use species and lineages interchangeably because you can think about lineages within a species, right? Different populations. What are the things, what do I need to understand to answer that question? What do I need to be able to build a tree that looks like this? Well, I need to know, is there an outgroup? We'll see very shortly why this is important. So it might not be kind of making any intuition currently, but right, if you're given a tree, you still need an outgroup. We'll define what that is. You need to, it's all relative. You've seen so many cases in this class where things are relative, right? Fitness is relative to the other genotypes. Dominance is relative to the other alleles. Traits that define how closely related you are as a species is dependent, is relative, based on what you're defining as an outgroup. So outgroup and rooted, kind of, they're very similar. We define tips, nodes, branches. We didn't find a root. This is a root. A root is also relative, right? It tells you, it defines that first kind of initial splitting event. It tells me that all of the organisms, the lineages in this phylogeny share a most recent common ancestor. Then there was a splitting event that created this diversity. So I'm going to look for an outgroup and a root, and then I'm going to look for when they began to diverge. Don't be fooled by the proximity of tips. Look for nodes, right? We're going to do some quicker questions that will test you on this. But, if I'm looking at this, Newt and Hart are more close on this graphical representation. This is just one way to draw this tree. So don't be fooled looking at tips. Look at nodes. So this is the beginning of how we're going to build an algorithm when we have a tree, not how we're going to build a tree. So tomorrow in class, we're going to build trees. So here's some practice. Here's a really big tree of different lineages of dinosaurs. So which species is most closely related to Shonisaurus? So I think this one's a word answer, the next one will be a multiple choice. So I have here a rooted tree, Ichthyopterygia is my root. So here's my root. You don't need to use the outgroup in this case. These are the outgroups. And then I have nodes. So where is the lineage of interest? Right here, right? So if you're asked a question about a species is an extant thing. Something, or it's like something that's represented as a tip. So I think a lot of you are putting a label that represents a branch. So this tree is different because we have fossil records, right? So you can think about relationships in dinosaurs that are contemporary, but you can also go back in time and actually find fossils that represent common ancestors. So for example, right, like this word is labeling that node and saying that all of the dinosaurs that way are part of that family. Like dinosaurs don't have families and orders the same way that species do, but that's what it is. So the best answer here is this one, right? Shastasaurus. It happens to be close on the tip, but what you should have looked for is a shared node. Okay? These two dinosaurs share a most recent common ancestor at the exclusion of kind of every other lineage. So what's the next one? So which species is most closely related to Stenotaurus? So where are we? Right down here. This is question 2. That was question 1. Which species is most closely related? So how are you thinking about this? What are you looking for?
The node.
Anyone want to say what they're looking at?
Which ones share a common node?
Yeah. So about 60 of you have answered, and 2/3 of you have not looked at the node. You've looked at the tip. So I think 40% of you, or what's 40 out of 60? Yeah, 2/3 of you put this answer. That's not the correct answer. They are the closest in tips, but you have to look at this node. Where do all of these species share a node, right? So your options were Ichthyosaurus, Brachypterygius, what was the other one? Platypterygius. So you can go here, here, here. Where do they all trace back with Stenotaurus? So this one. This is the node. That's the node that connects all of those lineages. So therefore, all of those other 3, options A, B, and C, are equally related. There's not one lineage that's most closely related. All 3 of them are equally related to Stenotaurus. Any questions on that? That's a really important thing moving forward.
The thing that threw me off was the other node that linked the other 3.
Yeah.
Or
there's a
the
lot
other
of
2.
nodes. Yeah, I agree. So there's a node here. You have to look for the node that kind of joins them all.
So even though there's another node after, it doesn't matter.
Yeah, so what does this node mean? This node means that Platypterygius and Brachypterygius are more closely related to each other than they are to Ichthyosaurus. But then this node means that all three of them are more closely related to each other than they are to Stenoterigius. So this is the node you care about. They're all equally distant based on that. So it's kind of like a nesting doll thing. There is a lot of hierarchy and relationships here, but the question was about this one. Right, so it's relative. We're seeing it's a relative answer. So why is that important to look at the nodes? Because you can draw trees any different way you want. Both of these trees are identical. These trees are identical. All that happened was a rotation of the node, right? All this is a graphical representation. All the tips are the same, all the nodes are the same, all the relationships between these two trees are the same, right? The order of them doesn't matter. What matters is that internal structure in the node. I can draw another tree that's exactly the same. Right? This would also be identical. I've rotated around these nodes. Right? I've taught this in an evolution class. We had kind of like a model of phylogeny, like a literal physical thing. You could see the nodes turning. You'll see this in discussion tomorrow, though. You can build a tree. All of you will build a tree. None of your trees will look the same. Why? Because you'll have just rotated nodes. They'll all be the same answer, just with different nodes rotated. Is that kind of clear? So in the picture from the book, the node that was rotated was the wolf-human node, right here. Right, just flipped. I flipped a bunch of nodes. They're all the same tree. So if you were asked to draw a tree, you can draw it in many different ways. Maybe you'll be asked to draw many trees. Okay, so I know it was really brief. You're going to get a lot of practice tomorrow, but given a tree, what do? Identify an outgroup, identify the root, identify nodes to answer questions. What about building a tree? So that's where we're going to transition to now. Any questions on kind of using trees that exist before we build some trees? Okay. For different species, so this is the trio I mentioned really briefly earlier. For different species, you can combine them in kind of a different permutation. How do you know which tree is the quote-unquote best, right? So here are three species. There's three ways that you can represent them. Three unique. So these are not the same tree, right? These are not rotations of the node. The first tree, A, chimpanzee and gorilla are sister, and B, chimpanzee and human are sister, and C, human and gorilla are sister. So this one, I told you, was the consensus. Most of the genome looks like this. The other two are equally valid trees. How do you choose? You have to think about which characters you're going to use, phylogenetic characters. So this is going to be kind of a broad way to do it, but this is going to apply to both phenotypic characters and molecular characters. Molecular characters just have a couple caveats that you don't see in phenotypic characters, but the terminology is going to be the same. So we use phylogenetic characters to build trees. So a character is an attribute of an organism, lineage, species, whatever word you want to use. I can provide insight into historical relationships. Right? And here we're thinking shared ancestry. It can help us define those nodes that we're gonna use to think about interrelationships. Characteristics that are unique to a lineage might help us define a species, right? I know a bat is a bat because it has wings and a mouse doesn't have wings. But the fact that bats have wings do not help us with shared ancestry, right? They're a unique characteristic that defines a species, but they are not a characteristic that helps understand evolutionary relationships. So we think of traits, right? Physical traits in molecular phylogenies. These are nucleotide positions. I would say nucleotide identity at given position. So mutations in the DNA sequence that again you can look at to think about shared relationships. So here we only have 4 character states, right? And phenotypic, there could be lots of character states. We often try to make them binary to make them easier. Like, do you have fur or no fur? Do you have opposable thumb, no opposable thumb? We try to make them binary because it simplifies the process. For nucleotides, we can't do that because there's 4. So, kind of, are you A, T, G, or C at a specific position in the genome? So if we can define characters, then we want to identify how they change. So for a molecular thing, that would be an alignment of homologous sequences, right? So it's like, I drew this on the first one. We assume that this DNA is identical by descent, it shares a common ancestry in genomes, and then there's changes, right? Some changes that are the same, some are different, but you can use this to understand evolutionary relationships. How are we going to translate these data into a tree? There's kind of 2 different approaches. There's the distance approach. You can quantify differences and define a distance. So that's like I'm trying to think of a good analogy. It's not coming to me. You don't want to just say they're different at kind of this position, you want to quantify it. They're different at 4 positions, right? Then you can build a matrix. Right? We could compare the differences between all 4 of these sequences in a 4-by-4 matrix, and that would represent the distance, the shares and differences from one sequence to the next sequence. The other main approach that we're going to see is parsimony. So parsimony is the fewest changes that give you a tree. It's like the simplest is the easiest, right? Or the simplest is best. So that's how we're going to define parsimony. So if we're thinking about back to molecular changes, what type of changes might be best for us to use? Which ones are going to give us the most information about shared evolutionary history? So the question is saying in general, which type of sequence will exhibit the slowest evolutionary change? Why is that important? Well, let's pause for a minute. Why is slowest important? Something I skipped, I was supposed to say on the previous slide. Why is it important that things are not too fast for DNA sequence? So DNA sequence, we're looking at things like this, right? An A to a T is a mutation. Anyone have an idea of why we want that mutation rate to be slow? Yeah.
You know that it's stable.
Sure, yeah, that's a good idea. I like that idea. Yeah, we can be more confident that we know what we're seeing. because of stability, but I think that implies something too. I think there's something in your brain, you're implying something's happening. Whether you realize it, back mutation, right? If something's happening in a DNA sequence, there's only 4 possibilities, A, T, C, G. If you get mutations from A to T, but there's so many mutations that you get a reversion back to A, a lot of evolution happened and you don't know. You're gonna misinfer evolutionary relationships due to high mutation rates. So in molecular sequences, oftentimes people wanna use something slow, meaning a lower mutation rate, so that if you see a mutation, you assume that it's shared. So that's the other thing with a fast mutation rate, you can get a lot of convergent evolution, a lot of mutations that are not shared. So slow is really important for 2 parts, reversions and convergence. Those are things that would mess up your history. Okay, back to the question. Which of these DNA sequences is going to evolve most slowly? I think about 2/3 of you have answered, so I'll leave it open. Let's start talking about some of these. Which one do you guys want to rule out?
Don't introns go rapidly?
Sure, yeah, so you can rule it out for 2, yeah, there's a lot of other stuff about slow. Sure, introns might not have a lot of functional constraints. So they might evolve slow, or they might evolve too quickly that we can make accurate inferences.
Mm-hmm.
What else?
Pseudogenes.
Pseudogene.
Pseudogenes?
Yeah.
Do you want to rule them out, or you like that idea?
Rule them out.
Okay. So we haven't really defined a pseudogene yet, right? So what is a pseudogene? We kind of defined them. I said previously that duplicates can lead to evolutionary change. Duplicates can also, so a gene duplicated, genes that are duplicated can also undergo degeneration. So they might not be functional, they might accumulate mutations over time. So I'm going to leave that up here for now.
That's probably the answer.
So what about, so we have now non-synonymous, so those are changes that are gonna, mutations that are gonna change amino acids, and we have synonymous, changes that will not change amino acids. So yeah, this question, I mean, it's kind of, it's not a super fair question in the sense of, you know, what is slow? We can actually put an absolute measure on slow. Non-synonymous, they will happen, right? Like, this is where the relative part, I don't want to dwell on this question too much. Non-synonymous can be deleterious, so you won't see them, or they can be advantageous because they're changing something, so then they'll sweep the fixation. So I would actually rule out them, because it's not kind of like a consistent process. It's like boom or bust. The same with synonymous. Even though they're not changing the amino acid, there is a lot of selection there. So we have not defined pseudogenes. You're not responsible for this, it's just to make you think. A pseudogene is a gene that has to be functional until it can regenerate. It has a lot of sequence, it's a full gene. So you can just kind of pick up mutations slowly. So if you put something else, that's fine. The point was just to think about it. Non-synonymous or boom or bust, same with synonymous. Introns are way too fast. That kind of leaves you with pseudogene. Even though we haven't defined them super formally, I think there's arguments that can be made. If you argued something else, I'm fine with that too, just to get you to think. You do not have to remember all of these definitions. This is how your book is going to define character states. You're going to see these tomorrow, so it might just be useful to get used to using them, even though you don't have to remember them. So this is how we can map character state changes onto a phylogeny. Let's just say we know the phylogeny, and now we're mapping changes. So transitions from the white circle to the black circle. We can just put terms on these that we all know kind of what we're talking about. So, plesiomorphy, there's too many words, right? Plesiomorphy. This is a character that's in the ancestral state. How do we know it's ancestral? It's because of this ancestral node here. How else could we know it's ancestral? We could use this as an outgroup. So the outgroup is kind of the group that's sister to everything else. We didn't define an outgroup formally. I told you to look for one. So the outgroup is sister to all the remaining taxa. So there's two ways you can infer the outgroup. You can look at the node, the ancestral node, or you can look at the outgroup. So we choose an outgroup because we assume that it has all ancestral characteristics. So any changes that happen are going to be apomorphies. So an apomorphy is a change to the derived state. So do you have to remember plesiomorphy, apomorphy? No, but if you use it to help everyone just have a common vocabulary for ancestral versus derived state, it'll be helpful. What about a synapomorphy? So that's a derived character that is shared, right? You can see that sharing at that node. So the black trait here is a synapomorphy. It's shared with those two lineages. And then an autapomorphy is something that's not shared. Autapomorphies are the least informative for building a tree. It doesn't tell us about, right? If you just have one black tip, it could go anywhere on that tree. It doesn't really matter. It doesn't tell us about shared evolutionary history. So they're often not informative, either for a distance method or a parsimony method. And then the last one is homoplasy. These are kind of shared by convergence. So this, we were saying we know this tree. We know the tree lets map the characters. Homoplasy can be really problematic for building a tree because you might be tempted. Both of these kind of have the black trait. They must be closely related. Sometimes that can kind of throw off your tree building, and we'll see why in a couple slides. So when we're building a tree, we want to use homologous characters that are shared. So we're going to use synapomorphy. So tomorrow's discussion, one of the problems is not on animals, it's not on living things, it's on nails and screws. It's really interesting, people have found, and people who study learning have found that if you try to teach students about phylogenies with animals, they'll get it wrong because they think they know something about animals. So you use inanimate objects, and it's easier to think about synapomorphies. And then the molecular part is on phage. So it's a DNA sequence that are phage. But in either case, you can think about synapomorphies of screws and nails and bolts, right? What type of head shape do they have? How long are they, right? Those are shared characteristics that you can identify and kind of group different clusters. So what does a synapomorphy give you? It gives you monophyly. So monophyly, mono like one, phyly shared organ. There's one shared organ. And we've seen this, but I haven't used this term yet. Monophyly is relative. You can define monophyly at many different scales. So that's what this picture is trying to show you here. So we can define a monophyletic group between lineage 1 and 2, right? They share a most recent common ancestor to the exclusion of everything else. We could define a monophyletic group between 4 and 5. We can define a monophyletic group for the whole clade. So if you're asked about monophyly, or to define a monophyly, you need to give the context. So, you know, I hope you're seeing the different ways that you can be monophyly. Sometimes it's better to see it in a contrast with non-monophyly. All right, so on this side, we have monophyletic. All of those lineages share a most recent common ancestor. This one's non-monophyletic, right? There's a node here that this lineage is part of, right? 1 and 2 are not monophyletic. There's a node in between them that actually says that 1 is monophyletic with the other parts of this lineage. And this happens all the time in phylogenetics. For a long time, people were building phylogenies from phenotypes, right? Reptiles are paraphyletic. So paraphyletic means that there's kind of like an extra lineage in there, right? So reptiles are not monophyletic. They don't share a node to the exclusion of birds. What would it need to look like for reptiles to be monophyletic? It would have to be like this, right? Turtles, lizards, crocodiles, birds. That would be monophyletic reptiles. That's not what we see. Vultures are a type of bird. They're actually what's called polyphyletic. So they occur in kind of two different independent monophyletic groups. And that was due to kind of the characteristics of vultures. So is that helpful to see what non-monophyly looks like? Other questions on this? I know this is a lot of information, so I'm going to try to wrap it up at the end. Questions? Okay. So this is kind of not super important, but I think it's interesting. It's kind of like whether you say Pluto is a planet or not. It might depend on, you know, how old you are. Whether you know a group of animals called reptiles or not might depend on how old you are, right? So most of you probably learned that reptiles are a group. Now we know they're not, right? Now we know that birds, so this would be like the old tree, based just on traits. This is the new tree, based on molecular data. So now, you know, some classes are not teaching about reptiles, they teach about Sauropsida, right? So the group that it contains what reptiles and birds. So there's a lot of like name changing that happens, but it's purposeful. It's to try to make sure each name corresponds to a monophyletic group. So what's the difference between homology and homoplasy? So homology is just the general term, right? This is the general term. Two traits appear the same. Right? Structures may or may not be homologous, they just kind of appear the same, but then we want to add something. We want to add something. We want to say it's from a common ancestor. It's not just appearing the same from nowhere, it's appearing the same from shared evolutionary history. And the alternative then would be homoplasy. So it is a shared trait, but it evolved independently. So that's where the example of the vulture is going to come into play.
Like
So sometimes you
mutation.
can say parallel evolution, convergent evolution. Interestingly, you can have a loss. So I talked about reversion. You don't have to memorize this. This is just parts that can give you homoplasy. And for a long time, people who study bird evolutionary history were really confused about homoplasy with reference to these vultures. And it's how they were defining a vulture. So if you're not familiar with that term, a vulture is often defined as kind of this bird that feeds on dead animals. And it has this really interesting adaptation where it has no feathers on its head, right? Because if you're sticking your head into dead stuff, you don't want a surface for bacteria to feed on your head. So there's vultures that occur in the old world, right? Africa, Asia. And then there's vultures that occur in the new world, which are the Americas. They have this really shocking trait that they share. So everyone was clumping them into vultures, but that's not really the case. what's really true based on kind of molecular data is that there's two groups of both. So that was a case where homoplasy would let you infer kind of an incorrect evolutionary relationship. So what are some other examples of convergence? Eyes, right? Octopus, fish, crocodile, hippopotamus do not share a common ancestry of eyes. It was an independent evolution of eyes in vertebrates, so the bottom row, and then octopus or mollusks, right? So that's three independent evolutions of eyes. If you were building a phylogeny just based on eyes, you would misplace where these animals occur. I think insects are another independent evolution of eyes. Reversals are super common in molecular sequences. I'm not going to spend a ton of time on this, but you will see this tomorrow in the phage example. Reversals can happen, right? So here we have an ancestral DNA sequence. They highlighted A, then you get a derived T, and then the other example, you get this reversal here, right? They can look like a homoplasy. They can look like a synaptomorphy. How do you differentiate them? You have to know something about the root or the outroot. So here it's telling you, right, the ancestor was A. Oftentimes we don't know that, but we can infer it. We can say we know all of these lineages are more closely related than this other lineage. We'll use it as the outroot. So these changes are all relative. They're relative based on how you define it. But that definition is really important. So what about parsimony? So I know the transitions haven't been super obvious in this lecture, but here's the transition. We can build a tree, or we can understand information from a tree that's given to you based on tips and nodes. We can build a tree through molecular characters. We just have to be careful about things like homoplasy. But then how have all this data? What are we going to do with this data now? We're going to use parsimony. So that's where we are here. So parsimony, the gist of it is that a simple explanation is favored over a complex explanation. There is a lot of quantitative theory behind parsimony. How we're going to apply it in this class is just you count up the number of changes, which phylogeny gives you the fewest number of changes. So another way to refer that would be the least amount of evolutionary steps. And we'll see a couple slides about this. So this would give you the most parsimonious solution. Every phylogeny is a hypothesis. When we're dealing with hypotheses and like chi-squared, we have a way to evaluate it, right? Based on our chi-square table, our p-values. We have a way to quantitatively evaluate our hypothesis of the phylogenetic relationship through parsimony. So the metric that we care about is the number of changes, the number of evolutionary steps. You're trying to minimize that to maximize the likelihood of your phylogenetic hypothesis being true. So here's an example. So these are all different types of animals. And this is, again, the eye example, where you have independent evolution of an eye. So in the top one, let's just assume that they all had eyes. It was ancestral. And then there's some lineages that don't have eyes. So the number of changes here is what? 1, 2, 3, 4, 5, 6. 6 changes. If you assume that all animals had a single evolution of eye and then lost it. And sometimes there's good reasons to make an argument like this. It's hard to evolve things. It's easy to lose things. But that's not parsimonious. If we assume kind of independent evolution of eyes, the number of changes is 2 in this example. So this one would be the most parsimonious. Does that make sense? For your discussion tomorrow, you have to use all the changes. You can't kind of build a tree on half of the data. If you observe data, you have to use it. So sometimes you'll see a lot of changes kind of accumulating on a single branch. Those still all count in terms of how you're going to calculate the parsimony of your phylogenetic tree. So we've mentioned outgroup kind of briefly. Why do we need an outgroup? It's going to polarize our changes. What does that mean, right? It's one thing to say that, but what does it look like? So this is our outgroup. Right, and that's making, this is assuming this white circle is ancestral, and this one is derived. So that's all it means. The polarization just gives you like directionality, which way things are changing. You know, if I had a different outgroup, then this would become the derived state. So all of the changes subsequent to the outgroup are going to assume to be derived, and it's really dependent on what you choose as an outgroup. So just one last example. I'll make an announcement now in case I forget. I'm having office hours today instead of Thursday. They're at 11 o'clock. Normal office hours tomorrow for both me and Dr. Gruey. So please come talk to us about this. My computer's about to explode, so we got to go a little fast. You know, you might not think about this in your everyday life. That's fine. I think it does impact you, like I showed you in cases of like COVID evolution. But it impacts kind of other things you might just think about kind of historically, like how do animals evolve? Maybe some of you wanted to be marine biologists when you were kids. Now you live in landlocked Nebraska. But this was a question, like all of the rules that we're building now, like find a root, find an outgroup, look at tips, look at nodes, that's come through like just centuries of people trying to classify animals to learn about evolutionary relationships. And for a long time, like you might know now that whales are most closely related to hippo because you learned it in kind of grade school, but people didn't know that for a long time. So these are two hypotheses that kind of represent the evolution of the whale lineage, that they diverged from the rest of kind of animals with cloven hooves. I forget the scientific name for that. Or they kind of evolved secondarily back into the water. And so the trait that people use is this hoof morphology. So this is like a bone in the hoof. Right? Whales don't have hooves, but they still have this bone that's a homologous structure. So homology. This bone is present due to shared ancestry. And the morphology of it actually tells you something about when whales might have evolved. So if you look at this bone structure, it kind of gives you two changes, right? It can say that there was a change ancestral, kind of all of these land animals, or there was a loss in whales. So this would, under just morphology, this would be the most parsimonious tree. It wasn't until people started sequencing genomes of animals, I mean, people they knew this one wasn't right. They're like, this can't be true. But they didn't have evidence, right? If you're relying on parsimony as your statistical inference tool, you're stuck there. It wasn't until people started sequencing genes and genomes that they then started getting a really interesting pattern. So now this is kind of the sequences of molecular sequence, and you can find shared things, right? So position 142 is shared among all of them. So it supports whales belong in this group of kind of hooved animals, but then there's other shares, right? So these two positions here really support late divergence of whales. if you were trying to add up all the DNA changes to make whales come out before camels, it wouldn't be very parsimonious. You'd have to have way too many independent mutations. So whales early, that one hypothesis, gives you 47 changes. Whales late gives you 41. So still parsimony, but the nature of the data changed and gave you a very different hypothesis that is supported. Okay, that's it. That's my last slide. Try discussion early. It'll only help